Okay. Well, let me just. One of the hopes of this meeting is to identify some of these flagships and ultimately attract funders both from local governments or international governments like CCI or Gates Foundation, which will really be facilitating the data production.
What the presentation is on the presenters mode.
Let me swap it then.
Yeah, thanks.
How about now?
Yeah, that's good, thanks.
And I think there's cancer we've been talking about, and Zemin also gave excellent talk yesterday about infiltrating leukocytes and even myeloid macrophages. I think there's a lot of diversity in cancer and relevance across different parts of Asia, and it really spoke to me yesterday about how the numbers are it's growing but still limited, and how we can coordinate efforts across the region to generate very robust cancer data set. I think it would be very critical in respect to also generating relevant healthy reference of these cancer types and profiling immune system there. Diabetes has been largely not spoken so much. I guess a little bit challenging to study, although there's a growing evidence that immune system is critical in diabetes, such as macrophages in adipocytes, for instance, and how we can tackle this disease in Asia will be a very critical component. And keep in mind that we do have collaborators in the US who are Asian Americans, and I think that's one area that would be very fascinating, is how we can work with Asian communities in the US or Europe to sample some of these tissues for more regional environmental studies will be very interesting. Infectious disease, I think that has gained a lot of attention recently, of course, but I think this mainly affects also a lot of developing countries, and how we can support, provide tools, and centralize some of the efforts will be critical. Aging is more of an issue in developed countries, such as in Japan and South Korea. But as I mentioned, I think this is going to be an issue in many parts of the world very soon. So how we can preemptively target this. And aging could come in many different angles, right? It could be neurodegenerations to immunity to many other diseases that are caused by aging. So it's really hard to pinpoint exactly what we mean by aging and what we should focus on, but that's probably a matter of discussion of how when we collect data, for instance, can we collect not only the aging, but how can we measure aging in the samples that we collect, for example. Genetic variation of, these are just pure genetics, and how we can attributes to gene expression and disease, and also response to disease. So more precision medicine towards that angle could also be relevant, and how we can leverage the single cell technology to address this will be very important. So yeah, I just kind of drew this morning. It's a very rough sketch, but it's kind of a ways to see the trends, right? And what you see here in the x-axis is aging or time, and you have this inflection point where when you're healthy, but you acquire a state of disease, and that disease can be definition defined by cancer or diabetes or infection and so on. But there's this component of y-axis where I think immune system also plays a critical role in inflicting these disease states, and how it attributes or further propagates the disease. And then there's this z-axis here I put as genetics and diversity. And I just added this word comparative genetics or comparative diversity after listening to the editor from Cell Press. You know, it's not very interesting to just report on what's already been reported. I think once we start comparing data sets from, you know, this region versus the Europe or the US and seeing uniqueness and differences, I think that's going to be also very valuable and insightful for the region. So it doesn't have to be, but I think we can think of in terms of comparative genetics as well. Just something to keep in mind as we discuss this. So that's kind of what I wanted to share, and I'll maybe have other people in the group to speak more.
Thank you for the overview, Jay. Just to kick the discussion off, if I can share a list of topics we want to go through today. We have 75 minutes. As you know, usually we never have time to go through everything, but let's see what we can do. Maybe sharing the wrong thing, just a second. I presume you can see my screen now.
Yeah.
And yeah, I'll leave it in this mode. So, so Jay very nicely listed the potential areas of interest, and that's really the core of today's discussion. Okay, can we identify some common interests across countries in HCA Asia and hopefully Australasia as well, that we can work on together across countries as an international coordinated project. And at least our experience has been that if we can really identify something that's compelling, and so I'll talk briefly about how IDA came about, right? So we'd already had the HCA Asia meeting in Okinawa, I think, and organized by Piero and Jay, where we said we should do something together. Then we had the second HCA Asia meeting in Jeju, organized by Sun Young Kim and Wong Yang Park, where we said, okay, what do we do together? And in the discussions there, we realized, okay, this is an obvious thing we could do together, right? And it's a low bar for entry PBMCs, comparing across 3 different countries, initially 5 different ethnicities. Can we see how human cell atlas cell states might be different in these different populations relative to, say, a Western population, right? Fairly basic, one small step towards achieving the genetic diversity goals and equity goals and so on of HCA Asia, but quite doable and something that was a shared interest across the participants. And that was all it took, actually. And then I think getting the rest of it going was just logistics. We managed to get funding from CZI. So the goal is maybe in this meeting, so this is the 4th HCA Asia meeting, we can identify some such shared interest across some subset of our countries. And if we really can, and we have competent, motivated researchers to lead it and recruit other researchers in their respective countries, I really feel the sky's the limit, right? So the question is, what do we care about? What's important for our local environment? And what's the intersection of that Venn diagram? So, and then we can come to the other topics, which is, are the samples available, the tissue banks, which technologies will be used, which funding sources, and so on. But I would say these topics at the top are the important ones. If we can agree on what's at the top, right? And just say, yeah, don't worry, I can get access to samples. That's most of the battle won. So I listed IDA first because it's our existing flagship projects. It's just expanded to India and Thailand. We hope it'll expand to even more countries, and we'll hope it'll expand to immune cells that are not PBMCs, tissue immune cells as well. I think that'll be very important. But maybe and if anyone has some comments on that or would like to join and can come up with ideas for tissue immune mapping, that would be fantastic. And then, of course, genetic diversity is one of the reasons we started HCA Asia, and certainly the reason IDA got started. So what can we do to increase the genetic diversity of HCA? So even in one country, right? One country is not a monolith. There's a lot of genetic diversity in individual countries. How do we adequately represent that? How do you represent the populations adequately in individual countries? I think we should never lose sight of that. And then, of course, we come to specific diseases. So there are Asian cancers. Cancers are more prevalent in Asia. And of course, there's a lot of cancer researchers in Asia doing single cell. So that's an obvious topic. Diabetes, as Jay has so nicely laid out, is relevant, and infectious disease for obvious reasons. Aging, because many of our countries, many of our populations, their demographics are changing. And then perhaps iPSCs and organoids. iPSCs could be the PBMCs, the in vitro PBMCs, in the sense that in principle, anyone has access to them. And that could be something we could do for genetic diversity across various populations. So that's just the outline. And I really hope, I hope at the end of today's meeting, we'll have a narrowed down list of, okay, this is flagship project number 2. IDA was flagship project number 1. What's flagship project number 2? What's flagship project number 3? Who's potentially interested in driving and leading those projects? If we can come up with that, I think that's the win. Even if we don't finalize that today, if we can at least get people interested in starting those discussions, then maybe in HCA Asia annual meeting number 5, we'll crystallize those, but of course, hopefully before that. So yeah, and I'm going to leave these slides up here for a little bit. Or actually, maybe let me take off the slides again so that we can see everyone's face. All of this stuff I put here has been posted in the chat. So please take a look at that. And please use preferably the Q&A to post your comments. And while you're thinking about that, maybe we can go around the room and among the panelists and see who has, I don't know, maybe take a roll call. Jay, what do you suggest?
Sure. Al has a...
I just wanted to follow up on the AIDA. So you mentioned that now Thailand has joined, and that would actually be really good to hear how that has happened and how can we roll this out to other countries? What is it funding has become available? Is it samples have become available? How did that work?
Great question. So the immediate impetus was the funding call from CZI to expand. So IDA is an existing HCA Asia seed network, or it's a CZI seed network. And then there was a call for a grant supplement to expand existing seed networks to underrepresented populations. And so we submitted a proposal for that. And that, but I would say that was not the starting point. Okay, so we were talking to Partha and Varadom and Ponpan well before that CZI grant call, because we wanted to expand IDA. So that one just happened to be the trigger for the funding application. But in general, I would say we're trying to expand IDA, and we're talking to people, and anyone who puts up their hand, welcome.
And is the data being generated in Thailand, or is it samples being shipped to Japan or GIS or somewhere?
Ponpan, you want to take that?
So data will be generated in Thailand. So we happen to be working on single cell RNA-seq for quite for 3 years now, and mainly we focus on infectious disease, particularly dengue, which is a very important disease in Thailand. So working in close collaboration with cell attachment group, we established the first single cell transcriptome in Thailand, which is not an easy task because no one else is doing it. But finally we get it established, and we are currently running 10x and single cell and smart-seq quite routinely now. So joining the IDA is a pleasure, and we think it's not that difficult to reach the goal because we've been doing PBMCs for a lot of PBMCs now. And for sample access, we actually get in touch with and would collaborate with a Genomic Thailand project, which they already have plans to collect PBMCs and run whole genome sequencing. So to be a win-win situation, we collaborate with them and would share the overlap some of the samples so that we can have both single cell transcriptomes and also genomes to evaluate EQTLs and other things.
Thank you, Ponpan.
Thank you, Ponpan. And just want to highlight the last point that you made. It's such an excellent point. Is that please communicate with your local biobanks when they're preparing blood samples. Just make sure they do it in a way that can be processed for single cell RNA sequencing. Then you can really benefit and have that synergy of genome sequencing and RNA profiling. So this goes to everyone who are represented in the country. So thank you for raising that.
So can I add some point onto this? Because the Genomic Thailand project is still ongoing, and they started to collecting the samples, and it's not finished yet. So when we have the IDA project, we talk to the PIs of Genomic Thailand, and we kind of get your ethics forms, inclusion, exclusion criteria, and also sample processing, and compare to the existing Genomic Thailand projects, and try to recruit new patients, well, new samples that would match with both criterion. So, and we have to have amendment of the ethics in order to comply with both projects, which I think it's important to do that at the beginning. And we will process samples the same way as Shyam and Jay also doing in Singapore and Japan and also Korea.
Thanks for bringing that up, Ponpan. That's a key point. And maybe as a template for other flagship projects. So in IDA, we're making all our protocols available. We put it on protocols.io. And we also have a Google Drive as a kind of starter package of protocols and things you need to do to join IDA or once you join IDA. And hopefully the other projects as well will have that homogenization and standardization of methodology.
Do you think it's feasible to expand ADA to say 20 countries? So like ADA version 3 or 4, you've continued to expand because you've got all the framework in place, and then it's getting a locally established capacity in each of these countries.
I think it's an interesting question. Where do we reach diminishing returns? But 20 doesn't sound crazy. It sounds sensible to me, and I hope we can do that.
Maybe I may comment a little bit, and this has a little bit of, how to say, a limitation on our side, in which I think could be addressed and should be addressed, is the technology. So, in fact, actually, Shyam and us, we had access to potentially more countries joining IDA, but because we started the project with 10x Genomics, we wanted to stay with that platform, and that limited quite a bit of including other countries that do not have 10x Genomics. I think that needs to be addressed maybe in multiple angles, how we can work with 10x Genomics to provide the platform in a more affordable manner, and or because understanding two different technologies. But yeah.
That raises a fundamental issue of the technology that Shyam also listed. I mean, what's the reason for sticking with 10x? Does it have to be the same technology? I mean, at the end of the day, if you get the similar kind of, we're all trying, I mean, HCA is not just 10x, and the whole goal of HCA is to integrate all of those data sets from all different technology platforms, right? We don't need to invest in raising the 10x stock shares market ourselves, but not purposely.
I don't have any stocks with 10x.
I think this should be a point of discussion because the reagent cost can be lowered potentially by other platforms, and some countries may prefer to take that route. And as you said, if we really stick with 10x, that really minimizes the number of institutes, labs, countries who can join.
I think so. Although, I mean, maybe off the top of my head, what would be a cheaper technology that's scalable?
I don't know if others want to chime in, but I mean, what makes it affordable for us is the multiplexing. So you're pulling 16 different individuals into a single 10x run. And if you consider other technologies out there, of course, we can generate our own droplet technologies, which may perform similarly to multiplexing 10x genomics, but scaling to different regions may be a little bit more challenging of disseminating customized droplet platforms. So we consider micro-wells or others. If you can adopt that to a highly multiplexed platform, I think it is worth definitely looking into.
And not to forget, a very significant fraction of the cost goes to sequencing, so which cannot be reduced dramatically. So that whether 10x is the choice or not, it's not just the reagent cost. That I think that play a little bit of a role, but the most you can reduce by replacing 10x with other platform in terms of the total cost, maybe, I don't know, 10-20% at most, in my opinion.
With the multiplexing technology.
Yeah, exactly. So if you take the sequencing cost into account, it doesn't really reduce too much. So.
Actually, Chung and Jay bring up 2 points which maybe not everyone will appreciate here, which is that at least in AIDA, we're doing multiplexing. We're pulling 16 samples in one 10x reaction, and we're doing 2 reactions per 16 samples. So in effect, it's 8 samples per 10x reaction. So the cost of a 10x reaction is divided by 8. I was muted at some point. I don't know when.
The most important statement was mute.
Okay, great. Well, if you're not good at lip reading, so most of the, you divide the cost by 8, right? Because we do 16 samples in 2 reactions. So that's not a lot, and the sequencing cost is greater. So it's hard to find another technology that's too much cheaper than that. Of course, you have to buy the machine, but still, I don't think that's prohibitive. That said, there are countries that we've considered for AIDA, and then we said, oh, that's too bad, because if you join, you will have to ship the samples to us because you don't have a 10x machine. And so it is a barrier. And so Aki, you raise a very good point, right? Maybe others. So we took the view that we want to compare across populations. And if you want to compare across populations, you want to have the same technology, because it's not like we're not discovering new immune cell types. That's the job of people already doing that in the HCA. Well, what we're doing is we're comparing healthy samples as a baseline across populations. And that is very hard to do if this population has this technology, that population uses that technology. But I wanted to get others' inputs also on this cost, technology access. What do you think of one technology across everyone in the flagship project versus flagship project having different technologies in different populations?
Maybe I can ask Zemin, because I think, Zemin, you integrated several platforms for your cancer study. I don't know what your impressions are. in cross-referencing.
I don't know if Zemin is back. He said he has to take a shot.
Oh yeah, he needs to take a break. All right.
Yeah. So we have questions in the Q&A.
Yeah.
One of them is on the 10x situation. I'll just read that out. So regarding 10x, this is from Juan Felipe Ortiz. Regarding the 10x situation, I think it might be better to establish standardized data formats instead of standardizing technologies. Anyone want to respond to that on the panel? Sehan, wanted to bring you in?
I mean, that's what we mostly do anyways, to standardize across the technologies when we are doing the data analysis. So I don't think this is the issue. The issue is actually technology and having subtle differences, whether we are interested to consider depending on the question that we are trying to answer. So I think one thing we should decide on, based on like now the experience in IDA, what questions are we planning to address by creating these atlases. And so that actually could, if like, you know, when answering these questions, if like using the technological, like these subtle differences are not important, I think then we shouldn't worry about technology a lot, but it really depends on what we want to achieve in the specific projects. And the other thing I think we should consider is like, first is there a variation, and how much variation we have like among the populations that we are interested in. And we should actually do I think a pilot study looking at that and trying to like, you know, find out whether the differences are really big and like, you know, needs to be like, you know, considered, or could be like considered little, or like, you know, we can manage it like by like doing like, you know, applying different statistical methods.
That's an excellent point. What do we want to achieve? And that really determines whether we're using one technology or using different technologies. And what we want to achieve in IDA was comparing across populations, for healthy populations. But in the disease projects, it may be a different objective. You know, disease versus control. Maybe you can do disease versus control in different countries using different technologies, but you're inherently normalizing within each country. So then maybe it's okay to use a different technology. I don't know if anyone else.
Yeah, I think you really need to be doing it using one technology. And even with 10x, I mean, we've been through 3-4 different versions in the last 2 years. So every time you change version, you're increasing the number of genes. There's things that that are changed. So a stable technology that you can apply cheaply across as many samples as possible is what I'd be going for, if possible. I'd be banking additional backup sample. So if a new cheaper technology like the combinatorial indexing gets more broadly utilized, then you can just go and do the samples again, and it'll be at a fraction of the cost of the original experiment. Yeah.
Yes, banking is an excellent idea. We should, before you profile and even after you profile.
So if you wanted to follow the ADA model, maybe moving it on to the next sort of samples, what would the next, so if you can expand ADA, yes, but what would be the next sort of samples? And I'm wondering things like epithelial, like skin biopsies or something like that, is something that's easily accessible. You can do a punch biopsy. So have you guys thought a little bit about what might be doable in that space? Similar, easy access.
Yes, that's really the goal of this discussion. One of the goals, which sample type and which question. So skin makes a lot of sense, and it's relatively easily accessible, and it can have the same philosophy as IDA. You know, profile healthy skin as a reference, and then diversify to various pathological conditions. But if your skin is great, if anyone, so we have the list here, right? Cancer, diabetes, infectious disease, aging, IBC. So And then Al brought up skin. Does anyone else have another disease or sample type you want to bring up as a potential flagship project? Besides what's listed.
That's a lot already. So I feel like that any of those are great ideas. I'm more worried about the start from any of those. I'm more worried about some of the logistics. If we start pick anyone, just say let's get this going to see what are the ethical problems, what are the operational problems, what are the technical problems. I mean, so it probably is less. The choice of any of the diseases probably not as important as just working out all the kinks. Seems like. I think, I would definitely vote for cancer because I work on cancer, but any other things are fine. It's a matter of, I'm more worried about whether either a funding source, either a way to share data, what are the technologies to use. Those are probably more important than the choice of diseases. So I would imagine. So,
yes, regarding the next question.
Yeah. Regarding the technology, sorry, I missed out the discussion for 2 minutes because I had to take a phone call. So what's it? Is there a conclusion on technology? It seems like 10x is probably one of the leading platform. I don't know if there's either good alternative. Of course we don't want to just keep on raising 10x stocks, but but the issue is what are the good choices? Right now it seems like that's a probably more reliable technology out there. The consistency is probably more important, I would imagine.
Anyone on the panel want to take that?
Maybe I'll comment what we've been working with. So we've also tried the same, like another droplet system many people are aware of by Dolomite. It's an automated system called Nadia to automate the original dropseq protocol. And it requires more starting cell number, but the reagent costs are lower per run. So it depends, like if the sample is precious in terms of you don't have as many cells to begin with, it's not that ideal. Yeah, but I guess you could probably also, I mean, we haven't tried, but you could probably also do multiplexing of these and it could even lower the cost even more. It's a bit more manual, like hands-on time in terms of the people who are preparing the libraries for this. So it needs a little bit more training. It's not so as easy as the 10x Chromium controller. It's definitely easier for somebody who's really starting to just make it work. It's more reliable in that sense.
Yeah, I would imagine that anything that can give us more consistent results across multiple different groups would be probably preferred methods. So yeah, I also know of multiple methods that claim to outperform 10x, which is fine, but those are really much harder to operate. So that really means that different groups doing the same experiment will likely produce, very different results. That can be the kind of problem, I imagine. But I would love to see more technologies. Right now, we feel like we are also trapped by 10x technology because those are so easy to use. And they are relatively consistent, actually. But that's an advantage they have.
Well, I was also talking to Musa recently, who's been doing a lot more. What is it called? Well-seq, the one that you put the mold. Yeah. So as long as you get the molds, and then can probably distribute those. Seems like you don't need fancy equipment, and from what I hear, what I gather, it seems to work well.
We have many comments. Thank you for a great discussion. Many comments in the Q&A and in the chat. I wanted to get to those. I think Tommy was the first. And so his question is, actually, maybe I'll bring up the question about technology before I move to that, because Tommy's bringing up a different point. Ankur Sharma says that integrating different technologies has its own advantages, such as some cell populations are better represented in SmartSeq or single-cell NucleoSeq. That's an excellent point. So perhaps if we really want to do a comprehensive Asian atlas, we have to do multiple technologies. I think that's a very good point. And Callum Parr, on the same topic of technology, says consistency is good, but once you get into tissues, diseases, tissues or diseases, the cell types or samples may not lend themselves to the 10x platform. This is where maybe the Nadia, the Dolomite system could come into play. So in a sense, same point that Ankur is making, that if you expand the number of technologies, you're expanding the number of cell types you can confidently process. It's an excellent point, actually. Adds to the total cost of the project if you do two technologies instead of one. But Zemin has pioneered multi-technology projects, and many others have as well. So it's a good point.
Let me try to address this question on Q&A, where what will be an issue shipping samples for countries where 10x is not available. I think that's a very important question, and we've been talking a lot about this within the equity working group, and it is a way to kind of empower the local scientists to be able to create their own pipelines, to have access from patients or individuals to research lab, to data production and sequence, all within the local environment. I think that's a lesson we all learned from our past genomic era, where many of these genomic samples were shipped to the Broad Institute or Sanger Institute, and they kind of took all the genetic data and published without really attributing or acknowledging the sample provision. So we really want to avoid this and ensure that the data production is generated on site by local scientists. So yes, it is 10x is a limiting factor. So we do need to address how we can ensure that data is produced regardless of technology as much as possible. And so let me just speak from the HCA perspective. So training is something that we will also provide. So there are a lot of labs that are not able to initiate because they're very just new to this. There are working groups that will try to provide instructions or even training sessions, either on site or they'll visit to ensure that the scientists can conduct the research.
Thanks,
Can I
Jay.
also comment about this, like where to carry out experiments and sequencing? I think, if I understand correctly, like the idea of CZI and HCA is to also introduce diversity in the sense that also they want to put more funding resources to local communities that are not getting as much at the moment. So by sending able to fund those single cell as well as sequencing means you're obtaining that amount of research.
It's really developing expertise in their local community, and that can grow and expand the scientific community there through funding support. Yes. Alistair asked whether there's an HCA Asia webpage, I guess.
Yeah, I mean, we're talking about which ones we should do, and Zemin mentioned cancer, and we do cancer. There's lots of people doing cancer stuff. We're doing some skeletal muscle and rare muscular diseases. Having a sort of a list of areas of interest may actually help bring us together. I think for some of them, they're too boutique to be a large funding proposal bringing everyone together. But like ADA could be done everywhere. A skin biopsy could be done everywhere. The muscle biopsy thing, it can be done, but it's not easily scalable. So maybe, is there an existing HCA Asia page with a list of stuff? Jay or Shyam?
There's no, I think Yoshi, I don't know if he's here. There's a plan to make a web page from the main global HCA website. So there's a plan to do this. I think, yeah, we did make a template, but it hasn't been approved by
I mean, I'd even be tempted to start up some Google Docs or something where people can edit and add stuff in, because that's going to be much quicker than waiting for someone to approve a official website.
And I completely agree with your idea of so how HCA Global has they created a biological networks. So there's even an email mailing list, email address. So if you want to participate in this organ, just email this person and you're in. So I think having that kind of system for Cancer Asia, for instance. And for that, we need coordinators, we need leaders, and we'll be happy to step into the expertise here in this group to maybe coordinate that as well.
Yes, both great ideas. As you know, the web page is in process. Google Drive, good idea. Yeah, we should do that. I wanted to bring up Tommy's question. Essentially, his question is, how do we do this genetic diversity analysis if you have a Western reference genome, right? But you're profiling ethnically diverse samples. It's a great question, but it's not only a question for HCA Asia. It's a question for the GTEx consortium. It's a question for the SCE-QTLGen consortium. It's a question for anyone who's done GWAS studies or exome sequencing studies, right? It's a general human genetics question. And there's no easy answer, because it's not just about having an Asian reference genome. It's also having a gene annotation for that Asian reference genome. And a SNP, a DB SNP for that Asian reference genome, right? And a structural variant calls for that Asian reference genome. That kind of infrastructure, I think people are building it, and I know in Singapore we're trying to build it. It's not there yet. So we're still stuck with using a Western reference genome to do single cell research and genetic diversity analysis in Asia. But all the national precision medicine programs, they're sequencing their respective populations. Hopefully something will come out of that. I don't know if any of the other panelists has any insights.
I imagine this is less of a big deal than the genetic variation. So if you're studying SNPs, of course, the reference genome is not more critical. But for this HCA, I imagine most of these studies are based on the transcriptome, where you are able to tolerate a lot of SNPs, a lot of variations among different ethnic backgrounds. So having a different background probably would not really change the results very much, because you're doing the mapping mainly to quantify gene expression in many cases. And also RNA-based SNP calling or mutation calling is not as reliable, so people don't need to do that as much. So I imagine this in the short term wouldn't lead to any problems. I wouldn't really just view this as a very top priority unless later on the sequence-based analysis becomes a lot more important.
I think, okay, if you're trying to do an expression QTL analysis, the reviewers will ask you, what are you doing about read mapping to genetically variable loci? And we are trying to do expression QTL analyses in IDA. So I think it's a legitimate concern, and the way people usually address it is they try to map to both the reference genome and the alt genome and show that and only pick loci. If you say a SNP in an exon is an eQTL, then you want to confirm that read mapping bias to that SNP did not create an artifactual eQTL and so on. But there are bioinformatic ways you can do that. They're not ideal, but that's what people in the eQTL community do generally. So it can be, it's not like you cannot do the science, right? It's just that having a local reference genome will make your science a little bit easier, a little bit better.
Well, can I propose something slightly different? I mean, we can keep on working on these problems, but then it seems like it's hard to piece things together of everything. I wonder, instead of thinking about what are the potential projects, I mean, can we think about if we were to make a statement of the HCA Asia team, this is HCA Asia project. So can we think about perhaps, what's our first paper is going to be like, for example, maybe in the coming year or two, we're going to say, well, we're going to publish a separate paper to show this is HCA Asia marker paper. What is that going to be? So maybe from that point, we can backtrack to see, what we need to do to get there. So otherwise, maybe it's harder to pinpoint. It's hard to resolve all the problems and the technical choices and reference genomes, and we can keep on discussing this for a long time. But if we think about, maybe a year and a half or 2 years, we're going to in 2022, we're going to publish our first HCA Asia paper or something. What is that going to be? What make that a strong paper? Maybe that get that focus on what we need to do. Then we can think about backtrack to see, well, if we want to show that Asian population for certain tissue has a different cell type composition differences against other populations, maybe that's a strong point or something. We can, if we think along this line, maybe we can help us focus on what's really needed for the first study. Then this can propagate into multiple such studies.
Yeah, and in a sense, that's like Sehan's question. Which problem are we addressing? Which question are we addressing? Thanks for bringing that up. And so that brings us back to our list of areas of focus as cancer, diabetes, aging, infectious disease. And so we've talked a lot about genetic diversity, so maybe we'll move on to the diseases now. For genetic diversity, Zemin, you're exactly right in either. We want to map QTLs. We want to say, how is this population different from that population, different from that population in cell type proportions, in gene expression, in genetic influences on expression. And it'll be that kind of survey. And I think if we do skin, we can do a similar question, ask similar questions for skin, healthy skin. But then let's move to the disease part now. So we have cancer, diabetes, infectious disease, and then we should talk about aging as well. And I want to bring up also Lipin Lu in the Q&A said, is there any interest in neuroscience related projects in Asia? Definitely, really depends on sample availability. So I don't know how, basically, we want to somehow narrow down a few priority areas, right? So Jay, you have any ideas for how to narrow it down here? And a few questions we can ask.
Yeah, I mean,
in flagship projects.
Continue on Zemin's point about what do we want to see in a few years' time. I think publication is one of them for sure, but also it's effort to just ensure equity is reached as well. So yeah, it's hand in hand that allowing scientists in the region to collaborate and work together to create synergy, which will lead to publication, but hopefully in return generate more data sets that will make more complete human cell atlas. Because right now I think China is leading a lot in data production, but there are other parts in Asia that could also be further represented. So how we can reach the level of equity in that and publish as a side. But also it makes sense from an organizational perspective and also to incentivize and motivate the scientists, how we can frame the question to more publication oriented. And I think I mentioned here earlier about like comparative diversity or comparative. So if you do find traits that are specific to Asian population that are not found in others, obviously that's going to be very unique. And that just kind of is a continuation of what the editor, right? The editor from Cell Press was saying. If Asian population generates a single cell data set of the liver, it's not going to be very interesting. And what new information are we going to gain from that? But if you do find, say, response to drugs in Asian population are different because the metabolism is controlled differently due to genetic variants, and we profile that across Asia versus European descent, that's going to be very insightful and useful for clinical purposes as well. So I think we do need to think also in that terms of comparative and genetics, and along with not just profiling tissues, but some more into metabolism or mechanisms.
I love Jay's idea. This is great. I think also along that line, if you want to start some project, and maybe we should not pick something that's very difficult. For example, you pick some tissues that need fresh tissue processing, lots of technical variability in terms of handling processing. Then you create a lot of problems. But if you pick something like blood-derived results, and for example, if you take any drug, like popular drug, like Keytruda or something, something like PD-1 treatment, before or after treatment, you look at the blood-derived immune cells composition. And if you have the data from multiple different countries, and that represent people from Thailand and Australia, Singapore, Japan, and Korea, and then this actually maybe this actually does capture a lot of what Jay has mentioned. I do like Jay's idea that if you take some kind of a treatment, you do some kind of before-after treatment, for example, then take something simple like blood, even though blood may not be the critical thing. Maybe it's less informative than tumor, but it's so easy to do that we should pick a low-hanging fruit, some kind of something like that.
I suggest vaccine.
Totally second this idea actually, because it's one thing to just take a snapshot of current healthy state immune system, but then to see how it responds. Obviously, we know with even COVID, like there's a different people responding to differently to the same virus, and that's leading to different phenotypes. So looking at the effects, responses of the immune system is actually, and blood, I totally agree, is obviously the easiest and the least variable and the easiest to interpret. So yeah, PBMC is definitely low hanging fruit, I think, and response to something. Yeah, I agree, it would be a good target.
Could not agree more. Excellent ideas. And I should say, we are trying to set up something along these lines for PBMCs in Singapore. And yeah, I would be happy to connect with any of you in the panel or in the attendees on something along these lines as an extension of IDA or as a separate flagship project, either way.
I mean, back to Zemin's earlier point about the logistics being very challenging, and it's completely 100% right. So blood is the low-hanging fruit, and through that we learned a lot and learned how to create protocols, IO, consensus-based questionnaires. So if anyone is interested in creating a group beyond immune cells, we will be happy to also provide the ropes on how to get started, because we had to start from scratch and it was quite time-consuming, but I think it's working now. So hopefully such platform can be expanded to other tissues. But yeah, blood is obviously the low-hanging fruit. But if there are other groups, yeah. Shyam.
I think it's a great idea to work with PBMCs and and blood and other cell types as well as like immune cells. But I think we should focus. I reckon, we should think about projects around funding opportunities. What projects can be funded in these countries? What are the, if you look at the epidemiology of diseases, what are the ones that are coming at the top? So we can actually target those and try to identify, find solutions to those ones. I think then there will be more opportunities to actually get funding and do research, build communities within the local environment, and also actually contribute more to the societies. At the end of the day, our aim is to serve to the communities within these regions, and how we're going to achieve that by focusing and prioritizing conditions that they are having problem with. So my suggestion is we said earlier, infectious disease is like, you know, one we know that, like, you know, is issue, or certain cancer types that are happening, or like, you know, diseases that was related to metabolism. So I think if we think of, like, even when we are studying the drug mechanisms, I think we should, like, you know, try to narrow down like our focus towards those. And the other thing that I wanted to make a point about generating or building up like research committees like locally, I think it's a long-term goal. Like, it's not going to be possible to actually establish like research environment within 20 countries that we are thinking. So instead of that, is it possible to actually create closed hubs? And so instead of like, yes, I'm not suggesting like one country or one side having like, you know, access to all the data sets, but having like, you know, maybe 4 sites where they create a network and work together. And I think we should learn from the past that, like Jay has like mentioned about the Human Genome Project, and like, you know, and create certain legal limits, and like, you know, which we will of course, like, you know, all abide, and like, you know, that shouldn't, like, you know, stop us, I think, taking the research like into next level. We should have learned a lot from the past as a all, like, you know, as a human community.
It's a good point, Sehan. There is a tension between being broad, you know, you want to cover as many populations as possible, and at the same time wanting to empower those populations and not say, give me your samples and I'll take them away. So I think it's really, that's part of the HCA equity working group's job, to say, where do you draw the line? How do you make the research work for the benefit of the communities that you know, that you're studying? And I think room for many more interesting discussions there. And I don't know if we can answer the question here completely, but I take your point. Maybe it's too strict to say we'll only profile the population if someone from that population can do the research.
Maybe comment on that as well. So I think hubs are great ideas. I think as long as there are no legal, ethical barriers to that, and also it comes down to funding. So who will support training and facility to invite, you know, researchers, and then right, it should be a training facility to develop the expertise and go back to their native country, and providing that opportunity is, I think, very, very interesting. You know, I think CZI or a lot of philanthropic foundation may be happy to support such missions, although that needs to be further discussed with them. But going back to your earlier point about framing our questions to funding opportunities, that's also an excellent point. And I think that made me think about industry. So we haven't really tapped into working with industry on this, largely because we want to stay away from stay focused on healthy tissues, or, you know, academic and industry becomes a little bit of a headache. And I think it will be a little bit challenging, more so than CCI, for instance. But, you know, the topics that we've been talking about, you know, drug treatments of the immune cells, comparative genetics, these are questions that industries are extremely interested in, right? Because they are providing the drugs, but they want to target the right population, the right groups of cohorts, and perhaps we can frame such proposals and work with industry partners to ensure that there are academic freedom and there are ways to publish and release data. And I think industries are slowly warming up to that idea. And maybe we can definitely explore this issue. So as an HCA Asia group, we engage the industry and come up with some framework that maybe we can help. We'd love to find leaders in that regard as well, framing agreements and coordination with industry. And I think there is a great need there.
So how does the CZI work? That's purely research funding without any strings attached. I mean, it seems like you contribute data. The data seems to be captured by Facebook. I imagine. I don't know. So yeah, everyone has academic freedom, but the company have access to the data as well. So if that's something acceptable, we can try to find additional industry sponsors or multiple sponsors who collectively support such a project. But then with the goal for us to use the data and publish data as we wish. At the same time, they have simultaneous access to the data. I mean, if we are fine with that, I'm sure multiple companies would be interested in that. And I'm aware of at least one that's willing to do this.
Yeah, so it's not an easy task. And, I think the global HCA is also tackling this issue as well in a greater sense. But again, going back to the question of genetic diversity and precision medicine and personalized medicine, I think we have a voice. I think we may have a platform to push for this more than, say, Western countries, for example. And maybe we can, facilitate this effectively. Yeah, I mean, it comes down to data, right? Like how to share the data. So I'm not CZI, so I don't represent CZI, but my understanding is the concept of DCP, which is the open data platform of HCA, is meant to be open. Right? So that means you have access, also the Facebook has access, or industry has access, so anyone has access to it. So in that, there's no barrier to there's no preferential access, right? So if the industry is open to that idea, or with minor conditions, I don't know, we want to limit the conditions as possible, much as possible. But maybe we can come up with a consensus platform that can satisfy the funders, including the industry partners. But I think that's going to involve a lot of discussion from both up and down the HCA organization. But again, I think from a genetic diversity perspective, we may have a stronger platform to say something.
I appreciate all the discussion, and I'd like to come back a little bit on the possible flagship project, because I think we need to reach some consensus here. And I agree with all of your suggestion, Zemin, Jay, Shehan, on the feasibility of the samples that we should consider. And also, I think we have to find a common interest as well, so that we also already have technical platforms and access to samples. And also, I agree that we should work on something that's specific to our areas in Asia, whether it's disease that most prevalent in Asia, such as certain infectious disease, or common disease worldwide, but Asian population has different response or maybe different severity of the disease, which I think taking all together the feasibility of performing certain projects in rapid time, and also intersect with this disease-specific and also common interest, would help us prioritize what flagship projects we want to go upon. And from what I'm listening, and at least from my team perspective, I think infectious disease, certain infectious disease, or certain type of cancer or cancer response might be what we want to aim for first, but I'm not sure. You can put in some more comments on that. But from Thailand side, I think infectious disease that's more prevalent here would be dengue, that we've already worked on, malaria, TB, and cancer. That we also have common cancer, but we also have country-specific or area-specific cancer, such as cholangiocarcinoma or certain type of hepatocellular cancer or cervical cancer. But we just started to working with Cancer Biobank and started to talk to cancer people. But I think for specific projects, we probably need to talk to our oncologist friends who have more like clinical insights of what's our difference between Asian and other countries, whether it's response to treatment or a certain subtype of cancer.
In terms of the infectious disease angle, different countries have different infectious disease endemic. I'm just wondering whether we could potentially group it by, so if we're looking at PBMCs, for example, and we're looking at PBMCs of infected and uninfected individuals from multiple diseases that are endemic to different populations, then you could say, okay, well, dengue looks like this and that profile is similar to whatever this other infectious agent is, which occurs in part of Japan or part of Australia or whatever. Is that sort of what you think? Because I think getting the same disease, infectious agent across all countries is going to be really hard.
That's a great idea, Al. And certainly in infectious disease genomics, people want to see the comparisons between this disease and that disease and autoimmune conditions and so on. You know, having disease A in country X and disease B in country Y might introduce a little bit of confounding. But hopefully, as long as you have a disease versus control, you reduce a little bit of that confounding. And so, yeah, I think if we could seed, I don't know if we can finalize today precisely which diseases we look at, but at least some great ideas are coming up. So, and then we'll come up with some suggestions at the end for how to take this discussion forward, right? But the goal is that we should have new, maybe seed networks. I don't know if seed networks is the right word because the CZI seed network grant call is finished, at least for now. But new networks forming around specific diseases or specific sample types that we can hopefully self-organize. For starters, I put in the chat the email address of the HCA Genetic Diversity Network. I think, you know, while waiting for the HCA Asia website and Google Drive and so on, please feel free to email genediversity@humancellatlas.org with your ideas. Say, I want to create a network on this disease or this tissue type, or I want to join a network on this disease or this tissue type, and we'll be happy to form the connections. But yeah, infectious disease is definitely of great interest. Cancer also, obviously, we haven't talked about diabetes. So as Jay summarized so nicely, Asians have different prevalence of diabetes, particularly in India, susceptibility, or even Indian ethnicity, people living in the US, diabetes susceptibility is tremendous. In Singapore, Singapore's diabetes rates are projected to overtake diabetes rates in the US, you know, within 10 years or something like that, or 10 or 20 years. So it's a big issue in Asia. It's not easy to get pancreas, but you know, there is an inflammatory component in diabetes, so blood would be very relevant. Yeah, so
to tissue immune cells, because I think again there's a growing evidence that say adipocyte-derived infiltrating immune cells or macrophages. So from a perspective of HCA, kind of stepping back a little bit from disease, but looking at profiling, and as an addition to IDA or branching of IDA is how about tissue immune atlas? So I think someone mentioned it earlier in the breakout session. How feasible is it to profile immune cells from tissues, resident immune cells? And I mean, we don't have to choose all tissues, but maybe a handful of them.
You will always get immune cells, right, from most tissues. I mean, yeah, most tissues. So it just depends, I think, on the availability of the tissues. If you can get hands on those tissues, you'll definitely be able to capture immune cells. And whether you want to just focus on immune cells by, you know, flap sorting them beforehand or not, that's a different question. But,
yeah.
Shyam, you want to comment on tissue access, healthy tissues?
Well, it's an issue, I guess, especially in certain like cultures. So I think we need to think that aspect of like the projects as well, and how we can improve and how we can approach the different communities, and consider their needs and and create a like, you know, more communication among us. I think for that happen, for like tissue sampling to be more heterogeneous across populations.
And have you had any success in, I know you have a massive PBMC project, it's really amazing. Have you made any attempts to access tissues?
Well, we in that sense, in Sydney, we are lucky at the Darwin Weizmann Centre. We have different projects, and we are working with the closely hospitals where we are actually collecting samples. Like we have lung projects that we're collecting lung cancer projects that we're collecting lung samples, brain tissue. I'm also speaking to some of my colleagues in Turkey. They are where they can collect lots of samples and they want to collaborate and they want to do single cell sequencing. But the issue is like they do not know people like us, and they don't know how they can create these like, you know, networks, and they are happy to like apply their local funding bodies as well as like, you know, international. So I think for tissue handling and like sampling, we need to be a bit more proactive individually with our local researchers and clinicians and start new projects and bring new people, like we have brought Ponpan to the team, and so we can actually like expand that and also create like new communities within the HCA Asia community.
Very much. So what healthy tissues, I know you can get cancer and adjacent normal, but what healthy tissues have you had success in accessing in a way that could support, say, an HCA Asia or Australasia flagship program?
I mean, when we are collecting disease samples, especially in cancer, we're also collecting normal tissues as much as possible, because you told me.
I mean, that's not healthy, right? So I'm saying not cancer patients, healthy people.
Well, the other thing is like, you know, is actually we created a biobank where we are collecting samples from people who are passed away, but like for other reasons, but they are happy to like contribute to research by donating parts of their bodies. So, which is
fantastic.
It's a challenge, but we have projects like, you know, we are doing that sort of work as well.
That's fantastic. Aki?
Can I just also mention, we haven't talked that much about aging yet, but that's also another like low-hanging fruit. So if we can, depending on the size of the samples we can collect, if we can get a, you know, wide range age representation, that's also like aging aspects, right? How it affects different aspects. So that also depends.
Yes. And PBMCs are the obvious thing for that. But also, like Shyam was saying, if we have a cadaver autopsy, whatever the term is, samples, that, you know, those will tend to be older, and yeah, that could be a source of tissue samples. In Singapore, it's not straightforward. At least we haven't managed to do it, but I think that comes to the issue of, you know, tissue access, tissue banks is we need a separate discussion on that in HCA Asia. How do we promote that? How do you promote organ donation? Or how do we connect up to the people who are already doing organ donation? How do we reach out to them and, you know, demonstrate that it's beneficial for them to contribute to this kind of science as well? I hope HCA Asia, my understanding is that there's not a lot of tissue banks and organ donation in Asia compared to, say, Europe or the US. And, but I mean, it's non-zero, it exists. And I think we should all reach out to these people who are involved. I think it would be very important. Ponpan, you have any comments on tissue access to healthy tissues? Or post-mortem?
Yeah, so we haven't done much of those healthy samples besides just healthy PBMCs as a controls. And other things we've done is quite similar to Shin, is adjacent tissue of cancer, probably, normal adjacent tissues. And one of our previous postdoc who's now leading her own group, Ananya, is working on dental pulps. So in the tooth. Yeah,
that's a good point. That's an accessible tissue, although I think mostly, yeah, I don't know if dentists can take it from people who don't have tooth problems, but.
Well, sometimes we have to extract the wisdom tooth, you know, that's quite normal tooth and healthy.
That's true. That's true.
That's
an excellent idea.
Mm-hmm.
All right. That's one more flagship project. We're running out of time. Jay, you?
Yeah, I mean, probably too big of a topic for further discussion, but hopefully plant some ideas in the future is the factor of environment. So we haven't really talked about potential role of environment and how, you know, yes, there is some genetic diversity, but also there's a greater environmental factors. And we talked about aging a little bit. And, you know, there are pockets of Asian regions that are more likely to live longer than others. You know, so is it the air that we breathe, or is it the diet that we're having, or, you know, the exercise that we're practicing? Microbiome is another issue. So it expands quite a bit, but again, it's potentially an interesting ways to collaborate across the region is the role of the environment. So yeah, I know it's too big of a question to start right now, but potentially a good seed network project.
Absolutely, absolutely.
So yeah, we have 3 minutes.
Yeah.
Is there a HCA Asia email address? Because we've got lots of people connected. We've been talking lots, and then we'll all disconnect and nothing will happen. So is there a way of us contacting everybody that's connected here today?
Yes. Actually, there is an HCA Asia mailing list.
Yep.
And we don't exactly know who, so we need to figure out who's in that mailing list and who's not. So because it came from the global HCA mailing list, and we're trying to subset the members based on where they're doing their research. So we would try to, so there's a lot of discussion whether that should be allowed or we should ask the individual permission to join this mailing list. So maybe we'll send out a email to all the participants of this HCA Asia meeting. Yep. And ask them to click here if you want to subscribe to HCA Asia.
Great.
So that they know they are joining the mailing list.
Or we could make it opt out, and suddenly we have 1,600 email addresses. If you want to opt out, you can.
Great. Yeah. Any finishing comments from Shyam? Go ahead.
I just wanted to add to your response to Zemin. I think we keep saying, yeah, we're waiting for approval for the website, for email and all that. It might sound weird, but we are part of the Human Cell Atlas. It's international organization, and we don't control it. And we want to fit into their policies, and we're saying we are a Human Cell Atlas Asia mailing list, Human Cell Atlas Asia website. We have to conform to their policies, and it has to be uniform all across the world. So in a sense, HCA Asia is on the forefront of this. We're the first people bringing up this issue to HCA. And so they're formulating policies, to make sure that works for us and works for HCA Latin America and works for everyone. Which is, by the way, HCA Latin America just started a month ago, inspired by the efforts of HCA Asia. So we are kind of setting a role model here for other regions of the world. Maybe there'll be HCA Africa in the future. So I think by exemplifying through flagship projects and showing demonstrations of successful projects, hopefully it will manifest to other parts of the world. So really encourage everyone to contribute and suggest and participate. Okay, well, on that note, maybe we can close the session right on time. Maybe I'd like to thank all the panelists for your insights and all the attendees and their questions. And we will get back, maybe Shyam and I will summarize, and panelists are welcome to provide additional inputs, and we will report back to the whole group at what time? And please email genediversity@humancellatlas.org for now with any ideas or proposals. So we'll report back at 3:05 Beijing time. And just as a plug-in, at 3:35 Beijing, there's actually a plenary discussion. So I think we will probably cover a lot of the things that we talked about in this room. What is HCA Asia and how to solidify the project. So hopefully, panelists and attendees, please be active in your contributions. Okay. All right. Thank you, everyone. Thank you all.
Thank you.
Bye.